This is the revision video for B1, disease and immunity. Infectious diseases are caused by parasitic microorganisms that infect you. These disease-causing microorganisms are called pathogens. The invading pathogen may damage cells and also release chemicals called toxins. How are infectious diseases and non-infectious diseases different? Infectious diseases are caused by microorganisms that invade your body. Non-infectious diseases are not. There are many different causes of non-infectious diseases, such as deficiency disease, if you do not get enough of a particular vitamin or mineral, disorders where part of the body is not working properly, such as diabetes, or inherited genetic disorders like red-green colour blindness that are passed on in your genes. Another example of a non-infectious disease is cancer. But what is cancer? Cancer is a term used for diseases in which abnormal cells divide without control and keep dividing to form an abnormal mass called a tumour. Cancer results in the growth of a tumour, but there are two different types of tumour. What is the difference between a benign tumour and a malignant tumour? Benign tumours do not spread to other parts of the body. They are easier to deal with as they can be cut out. Malignant tumours can invade and destroy nearby tissue and spread to other parts of the body. How can we change our lifestyle to reduce the risk of cancer? Some things we can do to reduce our risk are avoiding sunburn, avoiding eating too much fatty food and becoming overweight, avoiding too much processed food, avoiding drinking too much alcohol, avoiding smoking, and making sure we eat plenty of fruit and veg and exercise regularly. The diagrams show examples of microorganisms that can cause infectious disease. The picture on the left side is a bacterial cell and the picture on the right is a virus. There are four types of pathogen that can cause infectious disease. These are fungi, bacteria, virus and protozoa. The table shows some examples of the diseases they cause. One example of an infectious disease is malaria. Malaria is caused by a type of parasite known as protozoa. A parasite is an organism that lives off another organism known as the host, using it to get nutrition but also causing harm to the host. In the case of malaria, the human is the host and it is passed on from person to person by mosquitoes. Mosquitoes are what is known as a vector because they pass the parasite from one host to another, but they are not affected by it themselves. Malaria is more common in hot countries because that's where most mosquitoes are found, as they multiply more rapidly in hot climates. Mosquitoes also need water to breed, so high rainfall also increases the incidence of malaria. Malaria can be controlled by stopping mosquitoes spreading the disease. To do this, we need to stop mosquitoes biting people, and this can be done by using mosquito nets, insect repellent, and using insecticides to kill the mosquitoes. It is also important to drain areas of stagnant water to stop the mosquitoes from breeding. Antiviral drugs are used to treat diseases caused by viruses. They work by inhibiting the replication of viruses inside the host. Examples of antiviral drugs include Tamiflu and Relenza. Antibiotics are chemicals that kill or prevent the growth of bacteria. We use them to kill bacteria in our bodies when we have a bacterial infection. Penicillin was the first antibiotic to be discovered in 1928, but since then many others have also been found. Whilst antibiotics can be great in the treatment of bacterial infections, they have to be used carefully and should not be prescribed by doctors when they are not needed. It is important that patients taking antibiotics also make sure that they take the whole course, even when they begin to feel better. If they are not used carefully, it can lead to resistant strains of bacteria developing. An example of this is MRSA. So antivirals and antibiotics can be very useful in the treatment of infections, but why is it important that new drugs have to be tested? When new drugs are developed, it is really important they are tested to make sure that they are safe, to monitor the side effects that they have, and to make sure they can be properly taken up into the body and then broken down afterwards. It is also important to make sure that the drugs are effective against whatever it is they are being used to treat. The rigorous testing of new drugs involves testing on cells, laboratory animals, human tissue, and lastly humans, to make sure that they are safe and effective before they are licensed. Randomised clinical trials are used to see whether new drugs are better than existing drugs, and also whether they are better than a placebo. A placebo is a dummy pill and is given to a control group who don't know that they are taking a placebo, because sometimes people feel better just because they think they are taking a drug that will make them feel better. This is known as the placebo effect. Your body has barriers to stop pathogens from getting in. These include your skin, the acid in your stomach, chemicals in your tears, mucus in the airways, and the fact that if you cut yourself, blood clots to form a scab over the open wound. However, sometimes pathogens make it past those defences and enter your body. This is when your immune system will deal with them. You have different types of white blood cells. There are phagocytes that engulf pathogens, and lymphocytes that produce antibodies and antitoxins. Antibodies are proteins that can destroy a particular bacteria or virus. This happens because pathogens have markers on their surface called antigens. These antigens are different shapes which are specific to that particular pathogen. When your body recognises these foreign antigens, the white blood cells get to work. Each antibody is a specific shape to fit the antigen. Your immune system has to make the right sort of antibodies to lock on to that particular pathogen. Once a pathogen is coated with antibodies, the white blood cells can engulf it and kill it. Having immunity against a disease means that your immune system can destroy a pathogen by producing the right antibodies before that pathogen can make you ill. This happens when white blood cells have made those antibodies before. You can acquire immunity in two ways. Either you have had the disease before, or by vaccination. Vaccination involves putting a small amount of an inactive form of pathogen or a dead pathogen into the body. Vaccines can contain either live pathogens that have been treated to make them harmless fragments of the pathogen, toxins produced by the pathogen, or dead pathogens. Whichever of these is used in a vaccine, they all trigger the production of antibodies. This is then remembered by the immune system, so should it come into contact with that pathogen again, it can destroy the pathogen before it makes you ill. You are immune to this pathogen.